guest today. I am Shannon Cappellazzi, the lead project scientist for the Soil Health Institute, and I will be sharing with you a spattering of data related to nutrient additions from the North American project to evaluate soil health measurements. There is a picture here of me. I would love to hear from you if you have any questions or comments. There's also a picture of most of the 96 partnering scientists to help us make this project possible. I want to start with a thank you, and then I'm going to dive right in. So our task is really encapsulated in the title of this project. Our objective is to evaluate soil health measurements. And so we did this by going out to 124 long-term research sites throughout the geographic breadth of North America, which made up 689 different treatments and about 2,000 experimental units. We sent all of those samples off to 5 different labs to get a whole suite of indicators analyzed, which makes up somewhere around 136 measurements, depending on, if you count genomics as one measurement. Our amazing cooperating partnering scientists helped us compile a really thorough management history to make this absolute goldmine of a data set. Today, I'm going to try to focus mostly on the results related to the sites that had organic matter additions. As I think about evaluating soil health measurements, I always come back to the definition. There are a lot of different iterations of this, but at its essence, soil health is about the soil's capacity to function as a vital living system to sustain plants, animals, and humans. So to determine the best method, we always want to start with an understanding of what information we want. Our team discussed all of the stakeholders who care about soil health and what it is that they really want. And we came up with 2 primary ways that people might want to use a soil health assessment. And there are those who want to use a soil health assessment as an indicator of progress. They want to know if their management is making their soil healthier. And in order to do that, we are working on a program for soil health targets, which will be described in more detail in a talk by Dr. Christine Morgan at the upcoming Tri-Societies meeting. But we're also going to talk about today a little bit more. There are others who want to use soil health assessments to quantify functional outcomes. These questions are about whether the soil is performing each or all of its jobs to the best of its ability. So we're working on quantifying specific outcomes like the ability to store or cycle carbon, cycle nutrients, store or deliver water, filter water, suppress disease and pests, act as a reservoir for biodiversity, and likely more along the way. That's kind of where we're going and what we want to be able to do with that. And so now I'm going to get into just a little bit of the data that is specific to what we did here in our project. I would say that one of the most universally accepted points from the soil health movement is that soil organic carbon is important. Increasing soil carbon is not just for the sake of storing it in the soil, but also because that carbon is useful. It is used in the soil. The soil's specific capacity to function, to do that work, tends to increase when soil organic carbon increases, which makes some sense. Most soil organisms are heterotrophic. They need carbon for food and energy. If they have food, they can do work. And so we are looking here at a list of different indicators that we measured, just a subset of them, and we see that they all have this relationship to organic carbon. First one that pops out is this really strong relationship between total nitrogen and total carbon. It's no surprise, we know that these are coupled. But then I thought it was kind of interesting that soil organic carbon and total nitrogen have the exact same degree of correlation to potassium permanganate oxidizable carbon, which is often called active carbon. And then, as has been seen in other studies, potassium permanganate oxidizable carbon has the strongest relationship to the overall soil health score as calculated by Cornell in their system. So all of the indicators have a relationship to soil organic carbon, and it's actually relatively similar to that relationship to potassium permanganate oxidizable carbon. One of the ways that we have always analyzed whether or not a measurement should be used as a soil health indicator is whether or not they are sensitive to management. And so to do that part of this assessment, we used a meta-analysis approach called log response ratios. So within each site, we paired treatments that only had one difference. For this presentation, everything else was kept the same except they used organic matter in their nutrient management strategy, either as compost, manure, or biosolids. So using this method allowed us to measure the relative difference between nutrient treatments while we controlled for inherent features, allowing us to compare the reaction among multiple sites. So when you're looking at these figures, you want to note a mark that crosses the middle line means there's no significant difference when that management has been implemented. So for all of the carbon pool or carbon flux measurements that we analyzed, they all increased with the addition of organic matter. No surprise, right? You're adding a lot of organic matter, you're going to get an increase in carbon and carbon-related pools. Similarly with nitrogen, all of the nitrogen measurements except inorganic nitrogen measured with the H3A extract were significantly increased with the addition of organic amendments. So in the story of soil health I told earlier, soil health, soil carbon, soil functions, it seems maybe a little surprising that the overall microbial biomass using PLFA biomarkers did not show a significant difference, but we'll get back to that. A couple more of these response ratios. We tested just about every different indicator we had, and we tested every different kind of management practice using those response ratios. So here we're showing that measurements related to the physical structure and related to water did not have as consistent of a response to manure. So, the methods matter. Here we see that the wet sieve aggregate stability procedure called AgStab ARS on this did show a change in stability, while the aggregate stability method in the cache test and the phone app SLAKES did not show a significant difference. Bulk density did decrease with additions of organic matter. And then using available water holding capacity using intact cores did capture a difference, while the more standard repacked method did not. And so I think that, that's something, again, we're going to come back to, this difference between the ability to see differences using the available water holding capacity using intact cores versus the repacked cores. KFS, in case you're not sure what that is, that's saturated hydraulic conductivity. There was really wide variation. When looking at all of these sites together, we did not see a difference in saturated hydraulic conductivity, but often we did see differences between treatments at individual experiment levels. So in addition to responsiveness to management, we're also working on understanding inherent features, particularly related to soil forming factors and how those set a limit on the capacity of the soil to function. And so that's part of the reason that the geographic breadth of this project is so important. And so here, this map of North America is using the Hargreaves climate moisture deficit to color the map. And then you see those black dots represent our sample sites. The figure on the right is a detrended correspondence analysis of the 16S bacterial and archaeal genomic data from this project. So this is it's a multi-dimensional analysis and shows the two axes that explain most of the variation in this multi-dimensional space. The main thing to key in here is that the closer 2 points are to each other, the more similar they are. Each dot in that detrended correspondence analysis represents the entire community from one soil sample. So we see these samples from the most extreme dry climate in red, grouped over here, are very tightly bunched together. And most of the blue dots are bunched and with a gradient here in the color. And you may notice that there's a little bunch of these yellow and orange colored dots over here on the left. They may seem out of place. We checked it out. We thought that was interesting as well. Turns out those are in a dry climate, but in an irrigated system. I've seen a lot of these microbial community ordinations that demonstrate that pH is a driver of the microbial community composition. And in this assessment of our long-term agricultural research sites where pH is often managed somehow, it's no different. the color gradient changes along that x-axis, and that x-axis is what's explaining 45% of the variation. So we see that that pH is having is likely having a strong relationship here. And so this together, we're seeing that at the continental scale, we see that moisture and pH play a significant role in bacterial and archaeal community structure. Zooming in here a little bit, we have another detrended correspondence analysis, but now we zoomed into just one experiment in Kimberly, Idaho. It's on a silt loam with conventional tillage, diverse rotation, and no cover crops. We sampled 4 of their treatments. One that got 10 tons of dairy manure annually, one that got 30 tons of dairy manure annually, one with synthetic fertilizer, and one with no fertilizer. And while we didn't see any difference in the total biomass of the microbial community, this analysis clearly shows that there is a difference in the community structure. And so once we get to a small enough scale, we can see that the community structure changes by treatment. The axis, that x-axis down there explains 54.5% of the variation. And we see, those colors change from no fertilizer to synthetic to 10 tons to 30 tons. One more of these specific site-specific detrended correspondence analysis. Here we have an organically managed site from Michigan with sandy soil that used a diverse rotation and cover crops, and they had a factorial design using just organic fertilizer or organic fertilizer plus dairy manure compost and conventional tillage or strip tillage. And you can see by the color of the dots that adding dairy manure compost clearly caused a separation in the microbial community, but that the shapes, which represent the tillage types, are interspersed within those colors, and so they don't show that clear trend between tillage. There are a lot of other sites in the experiment where we do see a difference in the microbial community based on tillage, but here where we have the addition of manure and we're in a sandy soil, we see that the manure is driving the community change and not the tillage. So as we're working to evaluate the measurements, we continue to think about what we need to describe the soil's capacity to function. The capacity of the soil to make nitrogen plant available is probably one of the most obvious and influential functions for many producers. And so in this trial, we don't have a absolute direct measurement of how much nitrogen the soil makes available. But there have been a lot of studies that have related potentially mineralizable nitrogen to aerobic incubations of nitrogen mineralization, as well as those that relate potentially mineralizable nitrogen to field nitrogen mineralization trials. And they found that although it's imperfect, it's a good proxy. But still, there's no commercial labs that really want to run a 7-day anaerobic incubation. And so what we did is we used forward selection regression analysis, and we tried just about every different kind of sets of indicators to see how well we could predict PMN. The most predictive set of indicators was total nitrogen, carbon dioxide respiration measured with a 24-hour burst, clay, sand, and temperature. And so using those, that multiple regression for the row crop systems, we were able to explain 72% of the variation. And in the perennial systems, we're able to explain 85%. Using just the sites that had the manure added, we could explain 86% of the PMN response with these other simple indicators. We also have a full suite of functional metagenomic data for all of our sites, which is amazing. And I can't wait to relate what we found about these indicators to the specific genes that will really help us understand more about the microbially mediated nitrogen cycle. In the arid West, the ability to store and deliver water might be even more important to producers than nitrogen. And so our team has been working on new pedotransfer functions to calculate available water holding capacity using simple measurements. The current model used by most labs, shown by the red dotted line, is insensitive to changes in carbon. But our data showed a difference in available water holding capacity using intact methods for most management changes, and that was concomitant with a change in soil organic carbon. So the new model that we've developed shows a significant difference in the capacity to store water with changes in soil organic carbon, particularly at a low silt or low clay content. Interestingly, we don't see the same responsiveness to soil organic carbon in calcareous soils. Out west, we may have some more experience with this, and we know that calcareous soils have divergent behaviors. And so I'm particularly excited to use this inherent feature to categorize soils for this and maybe other calculations, but for this calculation of the soil's capacity to store and deliver water. I know I showed a lot of data here. Here are the main points. Carbon and nitrogen indicators are closely related. Carbon and nitrogen indicators are responsive to organic amendments. Physical indicator measurement methods matter. 16S community structure drivers of climate and pH were pretty strong, but then within a site, we can see that manure changes the 16S community structure. Nitrogen mineralization may be well calculated using total nitrogen and carbon dioxide respiration, and available water holding capacity using intact methods is sensitive to changes in soil organic carbon, but not in calcareous soils. So I can't thank our partnering scientists enough. Without their careful curation of these long-term sites and their help with sampling and metadata accumulation, this analysis would not be possible. And then I also need to acknowledge the generous financial support of the Samuel Roberts Noble Foundation, General Mills, and the Foundation for Food and Agriculture, who saw the importance of this project with this giant scope and scale.